Hi, this is Professor Tiffany Martin with Medical Microbiology. We are in Chapter 7, Microbial Genetics, and we had just finished talking about DNA replication in the leading strand and the enzymes involved with that. So the leading strand, we had the RNA primase come in, add the primer. We had DNA polymerase 3 come in, look for that 3' hydroxyl, add that in. We had DNA polymerase 1, take away the primer. So that is all happening, but we didn't talk about what's going on in this guy. Now, the same enzymes are involved, the same tools are involved. They will do the exact same thing. So let's do it on this side and see how that works. All right, so what comes in first? Yep, you got it, RNA primase. It's going to lay down a short primer on RNA. Then what comes in next? DNA polymerase 3. What's he looking for? He's looking for double-stranded followed by single-stranded. He's looking for that 3' hydroxyl. So let's find our 3' hydroxyl on my RNA primer. So I have 5' here, 3' here. So that means I have 5' here, 3' here. So where's my 3' hydroxyl? Right here. So DNA polymerase 3 is going to come in and he's going to add in nucleotides starting at that 3' hydroxyl, right? And he will continue adding in nucleotides. Which direction is he moving? As he's adding in, the replication is occurring in that direction. If I look, because of that anti-parallel, right, 5', 3', 3', 5', because of that anti-parallel, the leading strand, which direction was that DNA polymerase 3 moving? He was going this direction, which is following helicase. So in the leading strand, DNA polymerase 3 grabs on and goes. He will continue to follow helicase until there's no more DNA to replicate. That's why we refer to him as the leading strand. The lagging strand, same tools, same enzymes. They're doing the exact same thing, but because of that anti-parallel, it ends up going this direction, which means so my DNA polymerase 3 is moving along. But now I've got helicase, I've got more single-stranded DNA that needs to be copied. How do I get this strand copied? And I'll go back to one of your figures on this. If you look, not that one. If you look here, so there's my leading strand, my DNA polymerase 3 going through that. My lagging strand, the other side's going this direction. Helicase is moving this direction. As I get more single-stranded DNA, what needs to happen? How do I get this replicated? Well, the tools are going to do their same jobs. The enzymes are going to do the same jobs. We're going to have an RNA primase come in, put down a primer, DNA polymerase 3 come in, add in nucleotides, or DNA polymerase 1, remove the primer. But you see how this starts forming. Unlike the leading strand, which is like the Energizer bunny and just keeps going and going and going in one long peel, one long strand, this one is kind of forming in these little short segments. Now, these short segments are called Okazaki fragments. DNA polymerase 3 does a great job. He adds in all the nucleotides, but the thing he cannot do, his tool, the enzyme is not capable of doing, is joining the sugar phosphate backbone. Remember, sugar phosphates alternating? He can't do this last connection. He puts in the nucleotide, but he can't make this last sugar phosphate connection. So these end up being in fragments. So we need another enzyme, and this enzyme is called DNA ligase. And DNA ligase, to ligate, is to put things together. It's his job to come in wherever there's a sugar and a phosphate group that's not bound. He binds to it and connects it together. So most of the time it's binding all those Okazaki fragments. He does work on the leading strand. When the leading strand gets all the way back to the origin, DNA ligase will do its job there too. But on this section where we're getting all of these short little fragments, DNA ligase comes in and ligates these Okazaki fragments together. Okay, so again, it's the same enzymes are doing the exact same job, but because of that opposite anti-parallel orchestration, it ends up being a little different. You end up with a leading strand and a lagging strand. Okay, so some things I want to point out to you on some of these PowerPoints. This area right here, as it's being, helicase is separating this is referred to as a replication fork. And that's shown right here when this area, that helicase is opening up. So vocabulary term, this is a replication fork, and they're talking about the movement of the replication fork. And you can kind of see as those strands separate, the replication fork, the name for this location, moves down as well. All right, what other things do I need to point out? This is an anabolic reaction, right? We're building something. We need some energy. Where do we get that energy? Well, actually, these nucleotides that DNA polymerase III adds in are actually triphosphate nucleotide. Remember, we defined a nucleotide as a nitrogenous base, sugar, and a phosphate group. A triphosphate nucleotide has 2 extra phosphates, so it has 3 phosphates. We only need 1 to build DNA, and so DNA polymerase III, when it grabs a triphosphate nucleotide, it breaks off 2 phosphates, and that's going to generate some energy. And that energy is enough to add in and connect this nucleotide. So I wanted to point that out as well. All right, what else do we need to know? Do you know this whole process starts where? The origin. So the origin, again, just a sequence of DNA where these enzymes, helicase and other enzymes, will start to bind. But as it opens that up, there's single-stranded DNA on both sides of the origin as that opens up. So it ends up, these enzymes will bind to single-stranded DNA, right? So RNA primase. And so you end up with bidirectional replication. And this shows you how that happens. And you end up with 2 replication forks in that, and they will continue until they get all the way back, and then you get a termination where you run into double-stranded DNA, and it terminates there, and then you get the complete replication of DNA. So remember, bacterial DNA, all DNA actually, replication is bidirectional, and that's what that means. Okay, that was a lot of information, and this is where I would break you into groups in class, and you would actually talk back through. Do not skip this step. You need to talk this through. I cannot emphasize this enough. If you cannot get in a FaceTime, a Zoom with a classmate, you've got to talk it out to your roommate, to your mom. You've got to talk this out. It just really helps if someone can talk back to you that knows what this is doing, because this is too complicated to just go through and try to memorize steps. You've got to really talk it out. So make sure you do that, and then we'll pick up with the next video.